Okay, hi everyone.
Hey.
Hey.
Thank you for joining us today. I'm Ylva from Uppsala iGEM team 2017. And with me, I have Myra and Sanka, who are moderating this discussion. I also have a couple of friends, which is Misuse and Tinka. They are mascots for Uppsala iGEM Association. Hope you are excited about being here today. I thought we would start with a bit of an introduction so we can go around the table and everyone gets a chance to introduce themselves, where they're from, and what their project is about. So I can start by saying that Uppsala team is, we're trying to do a recombinant expression of alpha-crocin, which is a component from saffron. So we're trying to reduce the production costs. And it's going decent, like everything in biology, up and down, but we are having a good time. So if Zurich, would you like to just introduce yourself, your name and your project in a couple of words?
Yeah, so I'm David.
And I'm Irma.
So we are engineering bacteria that would grow in tumors and then deliver a drug in a very targeted and controllable manner.
And these bacteria are to be given intravenously to a cancer patient. So that's in short.
Okay, good. How's it going for you?
We're cloning.
There are ups, there are downs.
Yeah, no, we're spending
yes, of course.
Yeah, then trying to get all the clonings done.
Yeah.
So that's currently what we're doing.
Well, thank you. I'm glad to have you here today. What about Oslo? Can you tell us a bit about your project and who you are?
Hello, I'm Adel, and yeah, I'm Erik. We are currently trying to make a biolaser and implement it in yeast cells, and we're hoping to use that for basic research and diagnostics.
Okay, cool. And how's it going for you? Have you had a good summer?
It's going forward, is all we can say. This is hard.
Yeah, I understand.
Yeah.
Well, thank you for joining us. We're glad to have you.
Likewise.
To Lund, who are you and what's your project?
I'm Albert. I study chemical engineering.
And I'm Naomi, and I study biomedicine.
And what we're doing is we're trying to engineer a biosensor for detecting microplastics. And we do that by engineering E. coli and make use of the estrogen receptor and the NAHR receptor, and to make a logic AND gate, and that will detect the microplastics. And so far we hadn't had so much done in the lab because we've had problems with our making our plasmids. The company who was supposed to make them for us, they took longer than expected, and but now they're on their way, and we'll probably have work soon.
Yeah, well,
yeah.
Good luck with that.
Thank you.
And thank you for joining us.
Thank you.
So who else do we have? Is Granien, are you here?
So this is Flor and I'm Felix, and we're trying to engineer a bacteriophage detection system based on CRISPR in Lactococcus lactis, which is used a lot in the dairy industry. So that is what we're trying to target. And about like how it's going. I mean, it's going. We're doing stuff. So, but basically we have some issues with like when to order what, and then we forgot something, and then we have to wait for some G block to arrive or something like that. So we are still trying to figure out a lot of the day-to-day lab stuff, I would say.
It usually takes more time than you want to.
Yeah,
like
and also we all grew up in this environment where you're in a lab and everything is ready for you basically, and now you have to order everything and organize everything yourself, and that, I don't know, takes some adjustment, I would say.
So there's a lot of things that you're not really thinking about when you're only doing like horse labs.
Yeah.
Well, thank you for joining us.
No, we're happy to be here.
Good. That's everyone, right? Well, so I can quickly go through the rules, sort of. So we're going to discuss a few questions, and hopefully you all received them beforehand, so you had a chance to look at them. Otherwise, don't worry, you can go. You'll see how it goes, and like it's not that hard questions. It's just a discussion, so don't be nervous or anything. We will each take turns to talk. So I will announce the question, and then you each get a chance to say what your thoughts on the question is, and we will take turns. So you will respond with your thoughts and not reflect on what other people are saying. So if you have a comment for someone's response, you can maybe write it down for later. And then after everyone has gotten a chance to say what they want, we will have a discussion round where we will pass the word around. And if you have something to say during the discussion, you can write your team name in the chat and we will give you the word. Because during that time, everyone will be muted so that only person speaking will be heard. And I think that's it. If someone doesn't understand or wants something clarified, just write it in the chat. You each get 5 minutes per question to talk. And if you want to say more, maybe you can take it in the discussion time. But to start with, you have 5 minutes. And after that, I will have to cut you off so everyone gets to say something. So the questions are from a project that our team has been doing. It's an ethics book. So we've written a few chapters concerning different topics on ethics within synthetic biology. So today we're going to discuss genetic engineering. And we would very much appreciate feedback on the questions afterwards, because they are going to be used in this handbook, because we thought that we would do a handbook that every iGEM team could use as a structure and like a guideline for discussing ethics within their project. And we're ending it with 10 questions that we feel like every iGEM team should be able to answer concerning their project. So these questions are from the genetic engineering chapter. And we would like you to hear your thoughts, and if you feel like it's good questions, and yeah. So let's start. The first question is about uncontrolled release. So can we anticipate how our genetic engineered machine would behave if it were to be released in nature? Like, what would the ideal conditions for it to grow be, and could they potentially be met if they're outside the lab? Can we anticipate any interaction with any form of wildlife? So first, over to you, Sirich. What are your thoughts concerning this?
So I get most of the work with E. coli, and so are we. And I think the problem there is less that they, grow outside, and interact with plants or whatever, but probably more like when you, in the lab, you touch something and then you eat something which you're not supposed to do or whatever, and then you kind of get it on yourself, and then it might just grow in the gut or wherever, or in our case in the tumor if you have one. So I guess that's kind of where they might grow and where they might find a suitable environment for them to survive.
Yeah, but the thing is, so we are actually going to work with E. coli Misuse, which is a probiotic strain, and it's very non-pathogenic, so it really lacks stuff that some other strains of E. coli have that make it, so they make it less probable that this strain will actually survive. Even if it is, for example, orally ingested, I think there's very little chance that it will survive through the acidic environment of the stomach, etc. And so, yeah.
So now in our specific case, since we don't engineer any toxin or anything special in there for now, I would assume they wouldn't interact in a very strange way with wildlife, or in that case, you. So I'm not sure if it would be a big issue.
I would tend to agree.
Okay. If you want, you can discuss the question more in general as well, like outside your project, like what concerns could a synthetic biology have just in general? That's for all of the themes. Like you can take up both concerning your project and in general thoughts.
Well, I mean, in general, if you engineer stuff that is not naturally occurring and it gets out and it spreads maybe by to other bacteria, you might start spreading a new function into the environment, whatever that might be. And this might, of course, cause large effects on the ecosystem, on health.
Yeah. Good. Thank you. Do you have any other comments?
Not for now.
Good. Well, thank you so much. Let's go to next team. Groningen, what are your thoughts?
Since we're working with a bacteria that is used in the dairy industry, it might have the same growth conditions there. Usually, Lactococcus is used to ferment lactose into lactic acid, which is important for cheese production, etc. And since we're working with that, we could imagine that also living in there, since it's the same growth conditions. To avoid that, we will try to engineer a knockout mutant so that this mutant can survive without a certain nutrient in the wild. And yeah, I mean, of course, there could be some interaction with the local species, through horizontal gene transfer, especially since our idea would also implement the CRISPR immune system there, which is a huge selection advantage if you are constantly attacked by bacteriophages. So that could, of course, be a danger that this immune system gets transferred.
Good, thank you. Very, very good thoughts. Anything else you want to add?
Yeah, I think for us, it's not only our concern that it's for us realistically, it won't be a very big problem, perhaps, if it's inside the milk, or even though perhaps the survival rate of our own GMO won't be that high. I think the concern from the general public, if they hear that it is maybe in there, that is also a very serious problem if it happens. And for example, there's now a sort of scandal happening here in the Netherlands with a sort of compound inside eggs. And even though it doesn't, the chance that it's harmful to people, it's very low. But even though there's already a problem and all those eggs get sent back and some chickens get killed for it. And for example, if that happens to a company, even though the situation is different, it can be very bad for the company if that happens inside their factory.
I think it's also just a general GMO problematic. I mean, even if our system would get into a bioreactor and you would have GMOs producing your milk, it's not like we produce any pathogens or anything. They're going to be actually better at producing the milk because they have some immune system in place. But if you tell the general public, hey, we use GMOs to make your cheese, then they're all going to go like crazy about it, because people tend to be a little bit hysterical with genetic engineering because they don't understand it. It might be dangerous. So that's more the fear, I think, of us, that it's like a PR scandal if it gets into your tank. And the EU regulations also prohibit use of GMOs food production. So that's more like the regulatory issues, the bigger problem in our case, I would say.
Good. Nice. Anything else you want to add before we move on?
I think we're good.
Good.
Thanks.
Well, thank you. So,
Welcome.
Oslo, what are your thoughts?
Okay, it's working now, right? Yeah. Our project is, I'd say, relatively safe in that regard because we're only working on yeast cells and only infecting them with GFP, or transforming them with GFP. Again, that confers no tangible survival advantage, and the fact that we're trying to make the system into a laser also means that we need to have an external light source for it to work properly. So you're not suddenly going to see wildlife fungus start to shoot lasers at you, obviously. In general, at least for the genetically modified cells themselves, they shouldn't be a problem. The real problem, if there would be any, is, as Groningen pointed out, just general horizontal gene transfer, because even though, after all, we do have a plasmid that does have antibiotic resistance related to it. And I'm not actually quite sure how large the chance is that a bacteria should get a hold of that. In fact, I suspect that's minuscule. But again, we will have transformed an antibiotic resistant gene out in nature if we manage to let our cells get out. So yeah, that is something that's of potential concern. But other than that, no. Currently, it's relatively safe, and even if they would survive in a wild environment, then they wouldn't really have any advantage other than the fact that they're producing GFP, which, well, that's not an advantage, really.
Yes, in addition to what Erik said, we are using the Saccharomyces pombe that is categorized as a organism, microorganism with low without any pathogenic effect. So according to white list that is designed to classify the microorganism to 3 different levels. It's recognized as a non-pathogenic microorganism. So there is no effect on the human health or animal health. So cloning and transgenic yeast could not be any threat for human health. But we should consider in any case that transgenic organism could potentially have this ability to change the balance of ecological balance in environment. For example, if we consider the natural selection according to Darwin theory, those species that can survive, can match better with the environment, can survive and can be dominant over other species. So in general, we should consider also if our organism is not pathogenic, but we should consider that in the case of releasing in the environment, is there any possibility to change the balance of the environment? So I think it is important to consider these issues in connection to ethical consideration of genetic engineering. But in this special case, there is no in our project, there is no negative consequence for the environment.
I think we're out of time now. So.
Okay, thank you. Thank you for your response. And we will move on to Lund. What are your thoughts? Have you gotten a chance to think about the question? Sorry, we're unmuting you.
I cannot do this. Our technician had a heart attack.
Just a moment. I think that the grass team has joined us as well, so I can welcome you. Hi. So what we're doing is that we're discussing a question, and everyone gets 5 minutes to give their thoughts and their answer to discuss about the question, and then afterwards, after everyone's gotten a chance to say what they want to, we will have a discussion round. So then you can write your name in the chat, and if you want to say something, if you want to comment on anything that the other ones had said, and we will pass the word around and you can discuss. So apparently we're having some tech issues, but we're working on it.
You can unmute yourself.
Can you unmute yourself? Lund.
Yeah, we unmuted ourselves.
Okay, good. Perfect. So sorry about that. So what are your thoughts concerning uncontrolled release?
Well, the thing is that we're working on a biosensor that will eventually only be used in a lab environment. So we don't see a big risk of it going out into the environment. But if that would happen, then yeah, it will probably happen some kind of gene transfer, as a previous team mentioned, and maybe not the first year or the second year, but in the long run, something could potentially disrupt the ecosystem. And ideal conditions for it to grow is well, wherever E. coli grows the best.
Yeah. Well, may I add?
Yeah.
The thing is with our strain that we don't add anything that's toxic. We're just like the other team adding GFP or modifying one receptor, but there might be a problem with the antibiotic resistance, because when you grow cells, you use antibiotic resistance. So that would be the major problem if our bacteria would enter the world, because there will be an exchange of the antibiotic resistance gene or the plasmid to other bacteria, and that would be quite devastating. Yeah, that's no good.
And one of the interactions we could think of is, since we're going to use our sensor ideally in a marine environment, then a good way to anticipate its interactions would be to see how fish, for example, or other marine animal life would react to it. So yeah, that's what we have thought about so far.
Good, thank you. Anything else you want to add?
No.
No.
Perfect. Then we will move on to grass team. So the other teams, we did a short introduction round. So would you like to introduce yourselves and your project? Just a few words about what you're doing and how's it going. And maybe you can unmute yourselves as well.
Oh, sorry. We forgot to mute ourselves.
Thank you, Lynn. So.
Okay,
Now you're unmuted. So.
Hi. So
hi,
I'm Antonia.
My name is Tino. And our project is to control a robot. You can see the robot right behind here. It is mobile, and we use bacterial cultures to emit fluorescent signals. So we express GFPs of different variety. And we do this in according to a pH range, which the robot sends to the bacterial culture. And we read out the fluorescent signal and send it back to the robot, and the robot moves accordingly in an arena. So can I move on now to answering the first question?
Yes, sure.
Okay. Yeah, we have considered, of course, what we needed, what was necessary to use for this project. And since we are dealing with pretty basic molecular biology, we were able to use organisms that were established, and GFPs are also a tool commonly used in molecular biology, which does not afflict any damage to the natural environment should it be released. We have used 3 different strains for our project, and all of them are with some deficiencies, so they would not be able to survive in nature anyways. A risk that commonly can appear is that, of course, we have antibiotic resistances, and via gene transfer, these might be transferred to other bacteria if our bacterial strains should choose to leave our lab. But we have taken safety measurements, of course, and are working according to protocols to prevent the uncontrolled release of our bacteria.
Yes, and also as the other teams mentioned, there could be the problem of horizontal gene transfer, but also the things that E. coli is growing best at around 37 degrees is not that big problem here in Austria, as we don't have these temperature conditions. And you also have to consider for the growing factors things like humidity or pH. So in this point, it's not that big problem of any dangerous things. Yes.
Good, thank you. Do you have anything else you want to add?
Yeah, I think it's really a good strategy of the iGEM Foundation to prevent the use of dangerous proteins and stuff to just let people work in the lower safety safety labs and with lower hazardous materials.
Good, okay, thank you. Thank you so much, all of you, for your thoughts. So now we move on to the discussion round. Does anyone have any comments about what the other teams are saying? Let's get a discussion going. So if you write your names in the chat, we will pass the word around. So anyone has anything to add or comment about something that's been brought up? No? Everything's crystal clear.
Well, like, I think it's also, sorry. Well, like Groningen, I assume. So I think it's kind of hard to sometimes anticipate how it's going to interact with the environment, because there's so many variables, and maybe there's some horizontal gene transfer or not. So of course you can say, hey, they're not the right growth conditions, but maybe it will end up in some organism that can grow in your environment. And I think it's just really hard to predict. I mean, of course you can take safety measures to avoid that. But we all work with plasmids or something like that, so we all want to have to incorporate like a resistance gene or something like that, I assume at least, that and therefore they always have a somewhat growth advantage in the natural environment, since there are also lots of antibiotics there.
Good. Thank you. Anyone else has a comment?
Yeah, I would like to comment on the comment.
Okay, good.
He was talking about how the bacteria have antibiotic resistances, and these might be beneficial to growth in natural environments. And it's important that we use antibiotics that are not relevant to medicine anymore. We have used spigenomycin, chloramphenicol, and kanamycin. So I think it's irrelevant that there is a growth advantage, because these resistances are not that we don't depend on these resistances anymore.
But really, even in like food industry, etc., I mean, like let's say meat production, they use a lot of antibiotics that have not been used in the medicine for quite some time, actually. And you always have like residual compounds of still biochemically active antibiotics in the groundwater. So maybe that might be a problem, I think.
You want to add a comment to that, Ross?
Yeah, in this case, sure, it is a problem. I don't know if the antibiotics are used in the food industry anymore that we use in a lab.
They're
But
definitely not used in the medicine for any antibiotics. So.
Yeah, but resistances, if they are just on the plasmids, they are very likely to get lost over some generations of bacterial growth.
No.
Suraj, would you like to comment? Wait a minute, you are still muted. Okay, now.
So
now you can.
Yeah, can we? Okay. So we are using chloramphenicol and kanamycin too, but just a correction, chloramphenicol is actually still used in medicine. So this is kind of a potential problem. It's still used to treat certain kinds of infections.
Yeah, I'm not entirely sure, but a lot of these antibiotics are structurally very related, or at least functionally. And then from an existing antibiotic resistance, you might quickly evolve into something that is actually, or confers resistance to stuff that is actually still used. So I guess it's pretty important not to spread these antibiotic resistances, even though they might not be used widely anymore.
Thank you. Any comments? Anyone who has anything else to add? Okay, Norway has a comment. A second, and we will unmute you.
Yes,
okay.
Just in connection to what my friend said in other groups regarding antibiotic resistance, it is a major public health concern, and it is very important to consider the laws and the rules to ensure that the research, particularly in the field of genetic engineering and biotechnology, should follow the regulation that is approved by WHO, World Health Organizations, and hopefully the iGEM headquarters consider this case, and they have. I saw a very good link between WHO and iGEM in this issue. So every year the WHO, or every 5 years, it publish a new protocol regarding the high priority antibiotics and low priority antibiotics, and iGEM consider this in the safety form. So it is very important to decrease the antibiotic resistance in the future. If the research organizations and research institutes consider the public health rules that is approved by WHO.
Thank you for your comment, Oscar. If someone has something else to add, write in the chat. And otherwise, I guess we will move on to the next question. Okay, let's take question number 2. It's about misuse. Can we think some steps ahead and imagine a potentially harmful usage with our open source genetically engineered machine? So, Sirich, you have the word first.
Yes, so I thought about this question, and the thing is about our project, I think it's ethically controversial from the point of view that we are giving a bacteria to the patient, so for the patient. But when I thought about the potential misuse outside of this, honestly I didn't come up with any ideas.
Well, I mean, since we are trying to produce a drug or a treatment, there's always the danger that someone doesn't use it correctly. So, I mean, we're trying to inject bacteria into the bloodstream, which is a bit adventurous. And so if this is not done really right, then this, of course, might cause a lot of problems. So I think in terms of misuse, that would be specifically to our project, would be the biggest threat. Of course, I mean, we generate, or we hope to generate some understanding or knowledge of these systems that we characterize and that we plan to use. And that might help other people to build stuff that would actually be harmful or that could actually be misused. So I think the danger comes more from this perspective.
Okay. Thank you. Do you have anything to add? No. Let's move on to Groningen then. What are your thoughts concerning misuse?
Well, for our project specifically, we were thinking about just with how it's now, and if it's going to be used in a factory, on the factory floor, then, well, it's maybe a bit far-fetched, but then, for example, a person working at the factory want to cause the company some harm and just throws our bacteria inside the milk. And yeah, that of course has effects on the company and perhaps regulations or fines that they get by that. But that's maybe something, in a way can be harmful for the company itself.
Yeah. And we were also thinking, I mean, we engineer a immunity system, or we transfer an immunity system. And even if it is not per se dangerous in our organism, there's always the possibility of horizontal gene transfer to a similar species, which might carry pathogenic factors. Therefore, you could imagine this that you engineer a more resistant bacteria. So that of course would be a problem. But also just generally, we thought about some like problems with the whole open source iGEM project. And I think the simple fact that you standardize the protocols and basically try to make building a synthetic organism like Legos more a little bit complex, maybe makes it more accessible to more people. And if you are somebody with a mischief in mind or some bad intent, you could think, for example, of the use of a gene drive to which originally was thought to maybe sterilize flies or something that carries pathogen, and then use that to eradicate a species. So, example, if you eradicate a species that's a food source for a lot of organisms in an ecosystem, that could have really dire effects. But then again, if you really want to cause, as a terrorist, for example, harm, it might just be easier to blow up a building or, I don't know, get some weapons. So we don't really know how our system would have that much of a bad impact. But of course, you could think of a lot of things. For example, if you make a bacteria that eats a certain nutrient, which it normally doesn't consume, and therefore use that as a weapon in a war to deplete the food stocks of your army, or of the foreign army of the enemy. But yeah, I don't know. That's maybe a little bit far-fetched, you know. Like maybe it's just easier to blow shit up than if we're going that way, right? I think that's it from our side.
Yeah. Okay. Thank you so much. Let's move on to Oslo. What are your thoughts?
So we're working with lasers. So I know everyone here is already thinking about, well, weaponized lasers. This is not a concern in the immediate future because we need to have this stuff in specific setups to actually excite properly focused light. Does answer us to think some steps ahead, and if we're thinking quite a lot of steps ahead, then yes, this could become weaponized in some way, shape, or form in the future, conceivably. But again, this is one of those things that's so far off that other than actually providing insight on the subject, I'm not particularly afraid of how it could be used, or rather misused, in the near future. As for the subject of misuse of this project in general, I had a lot of thoughts on it, and they just got covered by the two past themes, in that yeah, it's completely possible to engineer an organism to do something harmful, both in terms of just being directly infectious or to deplete resources in a way or another. But you also have to ask yourself, is this more practical than you know, going for a more direct method to not use Groningen's words? Yeah, I don't think there's very much more to add, at least from my perspective on that. So,
okay, thank you, Oslo. We move on to Graf. So let us hear your thoughts about this.
For our project specifically, we don't see any potential that it causes any harm. We could conceive of a way that this small robot we see behind me is blown up to some deaf robot with lasers and something, but there would be easier ways to construct such a thing. You don't need bacteria in order to get a deaf robot going. But as other teams have mentioned, of course, the democratization and modularization, the simplification of biology has great potential to profit bioterrorists who might perceive of some viral agents. And this is a problem we will have to face and will have to think about, and therefore the regulation of the biobrick system and other parts of the do-it-yourself biology are important and should be discussed.
Okay. Thank you, Graf. We move on to Lund. Let's see your thoughts.
Yeah, you can hear us?
Yes.
Good. When it comes to GMO or gene modification in general, there is always going to be controversial things. But when it comes to our bacteria that we use and the things that we are modifying, the ER receptors and stuff like that, we don't think that it can be used for any bad purposes or like to make harm or anything like that.
No, because our target group for this sensor is mainly scientists who want to map out the levels in, for example, a small pond or ocean, small ocean. And so I don't think it's in the interest of misuse, to be honest. But since it's open source and anyone can take it and change it, well, then there's a potential for misuse. For example, we're working with the estrogen receptor, which is like a hormonal receptor that is very important in our bodies. And of course, from that, you can spin it off to make it to modify it and make it do harm in some way. Yeah, that's very unlikely.
Yeah, definitely unlikely that would happen.
Okay, thank you, Lund. Now that everyone's gotten a chance to present their thoughts, let's start the discussion round. So, does anyone have a comment about something that's been brought up? If you have something to add, just write in the chat and you will get the word. Okay, so
I kind of have just a general question for the Lund team. You're building an estrogen receptor, right? And you're trying to detect microplastics in the ocean, in theory, right? From ocean samples.
Yeah, exactly.
So don't you worry about false positives because of the whole xenohormone influx? For example, from the pill and from leached from certain plastics and stuff like that. Isn't that you get a lot of positives if you just have a estrogen receptor?
That's why we're building a logic gate that will, this is how it's going to work. We're going to have the estrogen receptor and another receptor called the NAJR. And the estrogen receptor, it binds to estrogen, of course, but it also binds to phthalates. And the NAJR, it binds to, or salicylate binds to it. And we're making since phthalates and inorganic, what are they called? Other organic pollutants. Yeah, exactly. Since they bind into these two receptors, and they also accompany plastics because they bind to the plastic surface and they're also in its surroundings, we can detect it in a two-way. You need to have both of these molecules to get a signal, and that's why the AND gate is there. So that will lead to a transcription of a GFP, like a 3-part GFP, and then in the end, we're hopefully hoping that they're gonna fuse together and give us a signal. Was that a good answer to your question?
Yeah, sure. Could you also imagine your system to work as a detection system for xenohormones in the groundwater, and so not detecting plastics?
Definitely, that could also be an application of it through some modifications.
Yeah. Okay. Cool. Just not ethics, but I was just curious.
Right. Okay. Thank you. Any other comments?
Oh yeah, yeah, we we wrote. I'm thinking like the thing with misuse is like synthetic biology and the how cheap it has become to like buy your own kit and make your own gene modification, that everyone basically can do it in at their home, and the risk with that is that, you know, for example, terrorists can create like super resistant viruses and similar organisms, and that's why, in my opinion, it's very important that we have some kind of like standardized regulation system, and that it shouldn't be so easy for everyone to obtain. Like we should definitely focus more on that in the community.
Yeah, I just want to say I totally agree with. Even though that it's an open access, you will need to have some kind of certificate to prove that you can work with it and to not have any other uses for it than, like, for example, in science or creating a biosensor or biotechnology. And not just that anyone can get a hold of it. But it should be easy in some way, but there should still be some regulation and some ground rules to follow.
Okay. Oslo, you had a comment?
Yeah, it's more of a question to all the other teams, really. Do you know, because it's related to what Lund were just talking about, do you actually know how the regulations are on, say, ordering the sequences, the genomes for viruses or stuff like that in your country? Because at least here in Norway, I'm under the impression that if you say you're a PhD somewhere, then you can just ask for the sequence and get it synthesized. So I'm interested in hearing your countries have been thinking about, or if you think it's something that we should be talking about at an international level, like in the EU.
Graf, you have a comment?
Yeah, I would like to comment on that. Here in Austria, I guess it's the way that sequences you order are checked, double-checked, triple-checked, if they are not containing any possibly dangerous sequences. And this is also done if you order just small parts of a viral sequence from different distributors. They will find you, I guess. But it will become a problem in the future when lab bench synthesizers might become available. And this is when we need to think about the regulations for these new technologies, since there might be the potential that this is hackable and will be misused.
Thank you. Groningen, you had a comment?
Yeah, so first off, I think you can basically order everything below ML1, you can just order. That's also been done in the biohacker community. So like people basically trying biotechnology at home. But if you want, let's say, if you want to have some AIDS virus or Zika virus, you actually have to have a certificate that you're working in an ML3 or ML4 lab, and you have to have all these regulations signed. So I don't think right now it's very easy. Of course, when you imagine when the whole do it at home kit, like a DNA synthesizer works at home, then it might be harder to just like since so decentralize the production to regulate it. But right now, I think it's pretty hard to, if you want to make a super virus, to work with it. But then again, like viruses are everywhere. If you know how to isolate them, then you in theory could like also work with it. But I mean, it's not as easy as I think people think to just build a super virus, because I mean, we're trying to build something really simple in contrast here. And I don't know about you guys, but some of our stuff just doesn't work out sometimes. So I think that might be an issue also.
Thank you. Lund.
Yeah, I just have a general comment regarding the how we store the genome and the plasmids. in the gene banks and stuff like that, because now I think that research is moving forward really fast, and there are a lot of discussion regarding how to store every kind of information. And I think due to that, we're moving so fast when it comes to progression for example, CRISPR-Cas9 and other stuff like that, which can be used for various purposes. And also when it comes to genoming the human and stuff like that, it's no one has created a really good way of storing this information. So I think that is the major problem at the moment, that no one has a really good idea how to store it safe so it won't be obtained. Because even though it might be difficult to actually like order something, like the previous team said, you can still get a hold of it just by using hackers and stuff like that. So, and there have been some problem with that before. And so yeah, I think the technology when it comes to storing needs to be improved, and then we might be safer in that regard.
Thank you. Would anyone like to make that comment? Does someone have something else to add? Okay. So we have a third question prepared if you would like to discuss something else. Wait. Norway. What's your comment?
Regarding misuse and the legislation, I studied an article regarding the legislation and the rules in the United States. So there is a no united law to cover these kind of materials, genetic engineering or so on the United States, and the different states, for example, has a different rules, and I think it is good to have a united law to cover this kind of research, because in future we don't know that what could be the outcome of, for example, GMOs that we are producing. So we should. I support the idea that one of the teams said that I think that making a certificate and providing certificate for the products that will release in future based on the genetic engineering, including for example, foods and etc., is essential. And for doing that, we need some basic rules, basic laws that is approved by each country governments and international level by the, for example, United Nations, WHO, World Health Organization, and so on. So I think at the moment we lack this supportive laws regarding GMOs and genetically modified organisms. I don't talk about the pathogens. Virus is special, and it is obvious that we should. There is no. It is not allowed to use. But also the non-pathogenic organisms should be covered by such rules.
Thank you, Oslo.
Thank you.
So if you had a chance to read what we just posted in the chat, we have a third question that we could discuss. It's about global justice. So what potential economic influence does your GEM have in its late stages of development and implementation? Do the economic and social benefits change if the GEM is applied in different countries? Are there potential sources of global injustice coming from iGEM? So have a think about that, and then we'll hear your thoughts. Sirish, are you ready to answer? Wait a second.
Can you hear
us?
Now
I can hear you. Sorry.
Okay. So I guess it's kind of the same with every new technology, that firstly it's the rich industrialized countries that profit from it. And since we are currently working on the therapeutic track, and especially there, it will definitely increase this imbalance that we in the industrialized world will have access to very potent new drugs and potentially cure things that in other countries are absolutely not to think of. And so we somehow need to make sure that what we create here also reaches the poorer countries, which often is very hard because, I mean, especially in our case, or when you think about all these new gene engineering, gene therapy things, that it takes a lot of infrastructure to actually implement this stuff. So definitely it's going to increase this this imbalance. And economically, of course, as well, because these new therapies will be very expensive to make and will pose whole new challenges to a health economy, which, again, we might tackle in the industrialized world, but other countries wouldn't be able to.
And then there's another thing. So the usage of our bacteria actually includes having an MRI machine and a focused ultrasound machine. And this is something that poorer countries don't necessarily have. MRIs are getting more and more common, but then you have focused ultrasound, which is something that's relatively new and won't be a standard part of a hospital for years to come. Which I guess is a valid question, a valid problem. And yeah.
Thank you, Sirich. Groningen, what's your thoughts?
Okay, first off, I mean, I agree with the whole that the first world will probably be the ones that implement these technologies first. But not to talk our project small, but we're like our project is more relevant in a larger industrial setting. I don't think a farmer in Argentina who has a small dairy farm cares about the amount of bacteriophage attacks. He just wants to have his product. And we try to implement it in a way that he can also use it. But I think the benefit is actually more for big companies that try to produce a lot of milk in Europe, in America, and in Asia. So I don't think we will spark a flame of injustice around the world with our project. At least we hope to not do that. And yeah, I mean, the benefits are for us more economic. You can produce more milk, you can produce more cheese, and maybe have more data on how frequent these bacteriophage infections occur in the dairy industry or in other industries that rely on bacteria. But yeah, I think generally we will not contribute positively or negatively to global inequality.
Thank you. Oslo, what are your thoughts?
So part of the idea with the biolaser we're working on is that it will be possible to use as a diagnostic tool. This has others who have done research on biolasers they have previously mentioned this as one of the options. And also hopefully that we'll find out that this is something that's relatively cheap and easy to implement in comparison to certain other diagnostic methods. If it is, and it's affordable to actually bring in the technology to other places, then this could be a useful tool in several parts of the world when it comes to hospital settings and diagnostics there. But again, that's provided that the technology ends up being cheap and straightforward. And the honest answer is we don't know if that's the case yet. And if it turns out to be useful but really expensive, then it's questionable how many places will be able to afford to implement it. And in that case, we're sitting on the case that we improved health in, or rather public health in the Western countries, but it won't be easy to export to other places and help that there as well. As for the implications as a research tool, again, this is one of those, will it be affordable? Will it be possible for universities in the third world or otherwise to actually implement this? Yeah, we'll have to find out and see what the price tag eventually is.
Okay, thank you, Oslo. Let's move over to Grass. What are your thoughts?
Yeah, I think with our project, it's kind of different than the other projects. They do diagnostic stuff that is directly directed to application. We were looking for something different, something that connects the information processing world and the biological world. And when we talk about synthetic biology, we really think about the synthesis component and not the biotechnology component that often gets associated to it. So we tried to create bacteria as information processing devices, and our molecular biology part just consists of pH promoters and also temperature sensitive promoters, which could be used in bioreactors. And of course, there's a chance that this happens first in industrialized countries, if it should happen. But we have to consider that we have a really new application, a really new synthesis of these both worlds that hasn't been seen before, and what evolves of this technology lies ahead of us, not yet to be known.
Thank you, Grass. Lund, what are your thoughts about global justice?
Well, it's always that when it's always the person or the company with the most money which have the highest advantage of getting things that we do. But we are creating a biosensor which will decrease the costs of measuring the plastics in the water. So it will be more available for others as well, in, for example, for countries and stuff like that, than what it is at the moment. But of course, it's always, as I mentioned, the person who has the money, who has the opportunity to buy it. But we're trying to create it so it will be more global than what it is at the moment.
And one of the social benefits is that if you introduce a very cheap way of detecting microplastics, you also make people more aware of the problem, and hopefully that will eventually lead to people becoming better at recycling and realize the harmful effects of plastic in the environment. So that's one of the social changes it can induce. I don't think there is some kind of way to create a monopoly of like our sensor, because it's something that is like
Quite general.
Yeah, it's something very general and something. How should I explain it? Yeah, never mind.
Well, as for our thing, it's not that high tech as for example, finding a new solution, a new cure,
for example,
or a new way to detect cancer, for example, when you need an MRI, as you mentioned. So hopefully, if it will be developed further, it's more easy to obtain it than what it is at the moment.
Okay. Thank you, Lund. Then we will start the response round again. So if you have anything to add, a comment or something that you're reflected on from the other people's thoughts, just write a comment in the chat and you'll get the word. Groningen?
Yeah, so I think that the general problem is that the things we try to design usually have a broad application and have a lot of applications in the third world. For example, monitoring bioplastics and curing diseases that are just not as present here. But you need a lot of infrastructure. You need a lot of knowledge, like professors that help design these projects in high-tech labs. So, like, of course, the iGEM approach just makes it more accessible to everybody because it's just easier to do. But you still need a lot of guidance, I believe. So I think that might be one of the big problems, that some people can capitalize on the gains of biotechnology earlier than, for example, people in the third world, where it actually would solve more problems than we actually have here in the first world. But that would just be my approach.
Oslo? Okay, Oslo, your comment?
Yes, well, I have a feedback to Groningen research team regarding the global justice and how, for example, increasing the amount of the milk and increasing the amount of fruit can benefit the farmers. Yes, it is correct, and if we look at this issue from this perspective, it could be beneficial for increasing justice and global justice. But at the same time, we should look at some intangible effect of GMOs or genetically modified organisms on other industries. For example, I have a friend that is studying on the effect of some toxins that is used for growing the fruits. So at the same time, we have more fruits because it is protected. The trees, the fruits is protected during growing from insects and other infectious agents. But his studies shows that at the same time the number of the fishes in the rivers is decreasing. So the fishers may less benefit from this kind of technology. So I think we should. There is many variables that play a role to consider why one new technology is helping to a global justice or not. Not easy. Sometimes we need time to monitor the effects of a new technology, and sometimes we can predict by some statistical analysis. Anyway, I think this is not easy to respond to this question, that how a new technology can help to global justice or not. We need to consider sometimes many factors.
Thank you. Would you like to meet that comment? Groningen?
Yeah, I think you have a very good point there, that it's indeed necessary to know the consequences of our products. And we actually also asked the dairy factory what would happen if, because of our products, they'll produce more cheese, hopefully, or at least they have less waste. And we asked them, well, if you make more cheese, do you buy less milk? And they told us that no, they won't buy less. So, well, of course, that's just one example, but I think it is indeed good to know about what they would do. And also for generally about other projects as well, that it's good to think about those things.
But like generally for our project, if you think about competitiveness, of course, in theory, our project, if it works how we want it to work, would it offer a competitive advantage to factories that employ this, because they could take countermeasures against these bacteriophages early on rather than later. But if you think about a company, let's say in the Netherlands, which produces a lot of milk with relatively modern machines, and like them outcompeting a dairy farmer that's just has a local farm and produces some cheese. I mean, I don't think our project would be the deciding factor, but rather what technology is employed, what kind of machines do they use to refine the cheese and process it. So I think it might offer like a competitive advantage to like big companies who have to like think about the economic implications, but not about like a small dairy farmer in the sub-Saharan desert or something like that.
Thank you. Do we have any other comments? Okay. Well, thank you so much for joining us today. I had a good time. I hope you did as well. We have a feedback form that we would very much appreciate if you would fill in. So we're going to post it in the comments. And so please fill that in and just leave any comments about what you thought about the questions and about the organization, if you enjoyed it, if you appreciated it, if you hated it and never want to do it again. Write that as well. It's good to know. And well, thank you. Thank you for joining us.
Bye.
Bye.
Success with the project.
Thank
Good
you.
luck to everyone, and hopefully we'll see each other in Boston.
Yeah, see you at the Jamboree, everyone.
Yes.
Yeah, take care.
Thank you for your time.
Thank you. Take care.
Bye.
Bye.
Bye.